from IIIT.
Thank you. Yeah, where is this? All right, so I think we are running a little bit late, so I'll straight away start my talk. And thanks the organizers, Chirag and Devnath, for giving this opportunity. So today I'll be sharing works that date back to 2017, when we started our lab, initially at ISI Calcutta, following IIIT Delhi. At that time, single cells were quite a buzz, but the methods were extremely slow. Although coming from Statistical Institute, I always could have gone down the statistical route, but I was so annoyed with the performance issues of the algorithms that were already there, like Seurat and Ranger at that time. Afterwards, a lot of methods have come, so I'm not talking about them. So we decided to take a very different off-road. Okay, so I'll try to give some intuitive idea about how these methods work, and this is literally using big data algorithms. So one of our contributions has been introducing big data algorithms in the field of single-cell genomics because it was needed. In bulk genomics, you can't use big data algorithms because the data is so small in number. But nowadays, people are using it quite frequently. All right. So I think the good part is the previous two speakers have already taken care of the single cell introduction part, so I'll not have to spend a lot of time. But essentially, this beautiful picture of different cells, quite identifiable from each other, get completely mixed up. You get a common signal, average signal, and you don't get to learn much from this, right? And this one analogy taken out from somebody else. It's not my own creation, but you see there are different kinds of animals, and then you have a unicorn. So none of these constituent animals are actually unicorns, right? So this is exactly what happens with the bulk RNA sequencing. And however, if you are able to look at a single cell RNA sequencing data, you can actually tell a lot more about the cell, how the cell is behaving. What is the difference between cell states and cell types? Not that every time you see a cluster, it's a cell type. It could be a transient state of the cell because it's just a snapshot of the tissue at that point in time. So we'll have to be a bit careful in that. But there are a lot of things that you can achieve using single cell data, as my previous speakers have already talked about. So I'll just quickly glide through this slide. Like you can see the cell type identification, the rare cell discoveries, the trajectory analysis to understand the dynamics from a single snapshot of the tissue. All these things are phenomenal and unthinkable that we are doing routinely today. If you see the Natures and Science of the world, they are publishing like 10 papers every month on single cell data alone without much of science, right? Well, so of course, if you have a lot of single cell data, you will learn something new. There's nothing interesting in that, but there are deeper questions that you can ask. I'll try to show a couple of such examples, not many, because in the end of the day, I'm a computer scientist, right? Trying to act as a computational biologist or a biologist, right? So there are different kind of analysis that you can construe using single cell data, are shown in this collage. So the trajectory analysis, cell typing, finding. These are the different steps of analyzing single cell data. Right? You'll have to find the feature genes, you'll have to do the dimensionality reduction, and so on and so forth. Right? We'll talk a few of them today. We'll talk about dimensional reduction, we'll talk about clustering, how to cluster very large single cell data in very small amount of time. We'll talk about rare cell discovery and a little bit of differential expression analysis, right? How we can write algorithms that really scale very large amounts of data. And one common theme in all of this is how you can learn massively without really learning. That is how you can learn zero-shot, how you can achieve zero-shot learning. You are not really estimating any parameters, but you are still learning something, right? So we'll show a couple of examples in that context. Well, so the problem of single cells is that it's very sparse. The gene expressions, like this is like bulk RNA sequencing sample, like you can imagine. So which is just a column vector. You have 20,000 genes in one sample, whereas single cells matrix will look something like this. You have a lot of dropouts, a lot more than what you can see here, right? And nobody knows whether they are down-regulated or whether they are dropped out. So yes, the noise level is pretty high in single-cell data. So you'll have to have very robust methods. You cannot entirely be dependent on statistical parameter estimations. So we'll get into a trap. Well, so I'll be talking about, so this is the landscape of the single-cell data analysis. There are a lot of new things people are doing nowadays, but you start with the raw matrix, you do gene and cell filtering for the good quality ones, and then you do normalization, batch correction, dimension reduction. Dimension reduction is you want to bring down the cells into the latent plane so that you can tease out the small variabilities that are given by different dimensions, so the different features of the genes you can think of. Clustering is absolutely important because that tells you what are the different cell type constituents in the data that you are presented with. And then you have pseudotype analysis that gives you the trajectory of how cells are transiting from one state to another state. I think Hamim does a lot of that. Rare cell discovery is essentially one of the big reasons for doing single cells, because 99% of the cells that you get to see in a particular single cell study are already well known. You know their markers. There's nothing new to learn from them. The only important thing is whether you can learn a new cell state or a rare cell type. And rare cell types are very important. For example, circulating tumor cells. They're extremely, extremely rare, right? Every 500,000 WBCs, you'll have one CTC. So that's one of the problems that we solve in our lab, which we'll not talk about today, right? But these are pretty much what I'll be discussing. The green colored ones are one where I'll spend some time today and talk about the algorithms that we have created. I'll not show you a lot of benchmarking results because, of course, we have published, so there must be something, right? So you can go back and check that. We'll talk about the ideas today. So hashing a cell is an idea which we really milked like hell, right? The hashing a cell means, just imagine there are two clusters. If you look at this room, right? So there's one set of people sitting there, another set of people sitting here. So if you just randomly cut this room, if you raise a lot of walls indignantly, irregularly in this particular room, then all of you will be segmented into small cubicles. But you will still be sitting with the same people that you are sitting, right? Even after doing this random segmentation, raising walls in the room, you'll still be sitting with the person you were sitting in most of the cases, if I do multiple times this operation. So that's a phenomenal thing that this Indian-born computer scientist came up with. His name is Rajiv Motwani. So he was the mentor for Sergey Brin and Larry Page of Google, right? Stanford prof. And his works are really, really seminal in the field of big data algorithms. So he gave the idea of locality-sensitive hashing. So what locality-sensitive hashing does is exactly what I just spoke of, which is if you have two samples and you draw multiple lines randomly, there are different versions of it, but what it essentially does is, if I randomly draw some lines, and if I project all of you on those lines, right? If you take, if you put a torch, and let's say your head is your data point, right? You are represented by the top of your head, so that's one data point. If I put a torch, and the projection that I'll get on that wall that I'm raising is essentially the data point that you are right now. So I can approximate you, even though I'm looking at 20,000 variables, 20,000 genes, I can actually very nicely approximate them with just 10 randomly raised boundaries. And how the cells are falling on either side of these boundaries would essentially define what this cell are. This is just like zip code. So how to bring down this big expression vector into a zip code was the idea. And that you can achieve using locality-sensitive hashing. If you think about it, it's a bunch of dot products which can be performed on a simple GPU also very, very rapidly. It's just a bunch of projections, right? You're taking your data points, which are vectors, you're projecting them on something else, some random walls, and that can be done very, very fast. No amount of thinking or if or else, nothing is needed. This is why I told this is zero-shot learning without estimating any parameters whatsoever. In this whole process, there is no decision that you are taking. But how does that preserves the identity and the similarity of the cells? Because machine learning revolves around, no matter how much deep learning, machine learning you cry about, in the end of the day, it is nearest neighbor, isn't it? Do you agree? Machine learning is nothing but nearest neighbor. You can do it in many fancy ways, right? So what it does is it's a very good nearest neighbor finding algorithm, where if you just look at this black and white boxes, this one represents one dot, That is, whether you are the left hand side, then I give you a black. If you're the right hand side of this 6 lines, then I'll give you a white. In this way, if I color code these data points, then you'll get, no matter how many dimensions within which these data points are lying, you can actually explain them with just 6 zeros and ones, right? A binary bit string. And if you find the Hamming distance of that, that is, if you just look at how many places you have a difference, like this is one case, then that difference, that Hamming distance, actually approximates the cosine distance in the original space, and there's a guarantee for that. So the moment I came across that, I thought that this is the key to all the problems of single cells, right? And we capitalized on that a lot. Well, so here it is given that if you have 2-bit strings like this, you can just see how many cases you have a flip, and you can approximate 1 out of 6 cases you have a flip. So that is the Hamming distance, and your denominator is the total number of hyperplanes, and that approximates the cosine distance very well. And the cosine distance in the gene expression situation is very good because it is an angular distance. It's not really the Euclidean distance, right? So there's a little more independent of any kind of batch effect. So this is quite a robust approximation of the single cells in a latent space. This is also latent space. It's just that there is no learning of the space, right? Well, so we sort of did a lot of things with this. As I said, the first thing we thought of doing was dimension reduction, right? That's a big problem, and it takes a lot of time. Even the PCA, not only a lot of time, if you do a principal component analysis, a lot of data, then what will happen is it will be swamped by, it will be completely dictated by the big clusters. If you look at the PBMC cells from a human blood draw, you'll see that T cells are 60%. And they'll have some other cells. Dendritic cells will probably be very small in number, right? Macrophages will be very small in number, right? And so on and so forth. Megakaryocytes will be probably even little in number, right? So what will happen is principal component analysis, the way it operates, the way it optimizes its objective function, it always tries to be at the consensus of all the cells, right? It tries to appease all the big cell types like T cells, etc. That's the problem. It takes time, of course. It is time consuming if you do the calculations. No matter you do TSNE, UMAP, or whatever, they are going to be troubled by the big cells, big fat cell types. And you will eventually ignore the smaller ones. So we did a small tweak here. So what we did was we introduced a structure preserving sampling kind of an idea. I'm not sure if in the field of statistics we will have come up with these ideas. I think people have. But I really didn't know about that, right? Later on, I saw some studies where people have talked about the structure preserving sampling in a very statistical sense. We didn't want to do that. We just wanted to find a hack for the problem that we are facing. You look at all these cells. So what we did was we used the LSH to find out the nearest neighbors, and then we randomly dropped points which are getting connected. That means they are very close by. So there is no incentive in keeping a point, right? If it is already if another person like that is already there in my data. So I just did that. So you take all the data points. It's so stupid, right? You take all the data points, look at the nearest neighbors, drop the nearest neighbors, start dropping. By the time you start the first 10 drops, a lot of the data points are actually removed from your data, right? And it is also structure preserving because you are removing a lot of samples where a lot of data points coming from the majority cell types, right? So once we did that, and then we did the principal component analysis, the regular principal component analysis, and then we did a post hoc projection of the removed cell types. So if you see, this is just a small number of cells which we retrieved or obtained after this dropping activity, and then we did a principal component analysis, and then we projected back the cells that I dropped, so I recovered them. But by virtue of doing this, I preserved the structures in the data, therefore we got better results. So here are some silhouette indexes that measures how well clustered and structured the data are in your principal component space, so we did that. It's a very fundamental, I didn't really want to go with, I didn't get into too many single cell analysis, taking different data and do this, do that. It was a very simple fundamental paper. It worked on few cases, we just demonstrated and that's it, right? And we used some tweaked version of that. I'll not discuss the results too much, but yes, it improved the performance like the ARI likes of what Hamim has already talked about, right? So there are measurements that you do, like how well it is clustering the cells that should be clustered together, etc., etc. So on all those quarters, it did fairly well. Okay? Now coming to the clustering part. So clustering is the most important problem that we got stuck to when the single cells started coming out in volumes, right? Like when this droplet sequencing methods came out commercially, like 10x's and of the world, then we thought that, okay, our clustering methods really don't scale that quickly. So what to do with that? So we also made use of the locality sensitive hashing here. We also came up with a different structure preserving sampling, and I'll try and show you like what it does. These are all PBMCs, 68,000 PBMCs. This used to be one of the go-to data for the benchmarking when I published this story in 2018. And it has 68,000 PBMCs, blood cells, and the grayed out ones are ones which are discarded from the sampling, right? The red ones are the ones that are taken from the sampling. So what you see here is like dendritic cells. So when you just take 500 samples, 500 cells by the structure preserving sampling method that I just described, if you just take 500 cells out of these 68,000 cells, that is a very small number of cells, isn't it? But within these 500 cells, you see how unevenly it got distributed. It is tending to come more from the clusters where you have smaller number of cells. Isn't it what you expect? What is the point in taking 2,000 cells from this guy? Because you have so many representative cells from that. It's so fat cluster. You want to maintain some kind of a democracy there, right? You want to take more cells from the guys who are minority than majority. And this is exactly what is happening here, which is in contradiction to what would typically happen if you just go down the random sampling route. If you randomly sample 500, then it will be all over the places. There will be no specific bias to the minor cell type. You understand what I'm saying? So as you keep increasing, then everything is everywhere, right? But when you take just 500 cells or 1,000 cells, then it is much better that you preserve the structures. You take more cells from the clusters which are which are minority in your data. Clear? Just you can ask me a question anytime. I'm going a bit fast because there are too many slides. Okay, but you can stop me. It's my nature that I speak fast. Just stop me when you feel. Yes.
So the distance that you use is Euclidean distance.
It's a cosine distance in a way. Yeah, because locality sensitive hashing approximates cosine distance.
So when you're removing cells,
yeah,
you are using that distance.
Yes, implicitly that distance is being used. So another idea, I'm just talking about a couple of intuitions. So how we get carried away by methods that are already there, like deep learning, machine learning, this learning, that learning. But every data demands some amount of thinking. I'll tell you, this simple old good method principal component analysis has a scope of improvement. Look at this case, right? This is the first principal component, right? This is the first principal component. What will have, there are two clusters. If you project these two clusters, the cells of the two clusters on the first principal component, which is explaining the greatest variance in the data, then these two clusters will lose their identity, won't they? Just imagine just projecting all these cells on this line. So the greens will be inside the reds. So you lost the vision, right? But if you look at the second principal component, it's much better. It's a very good example that I sort of personally came across. I construed this example to convince me that we need to do something about it, because whenever we would use the principal component analysis, people would say that, okay, take the top few principal components. So it's not as straightforward like that. You can also take the principal component based on whether you are seeing multiple modalities, those are getting formed after you project the cells on the principal components. For example, in this case, if you project these red cells and the green cells on this first principal components, you'll just get to see something like this. But if you project them on the second principal component, you'll get them separately. So no-brainer, right? We should do it. So there is no reason to go for the first principal component all the time indiscriminately. And this is what exactly gave us a better result. So here in this figure, we show that how many modes your principal component explains. This is the variance that principal component explains. Of course, the variance is very high in the first, and then a little low in the second, and a little low in the third, and so on and so forth. but the number of modes that are getting formed, which you can measure using Gaussian mixtures or splines or whatever. I'm not getting into the mathematics of it, but the number of heads that you can see after projections, if you measure that count, then you'll see that this is not always sharply falling as you go down the principal component numbers. As such, we chose, so you see the top principal components are 1, 2, 3. 1, 2, 3 are the top principal components, and these are some of the cells. These are some of the cell types, and you see that they are completely mixed up. Whereas if you see the component 2, there the cell types are slightly more spaced out. So component 2 was elected to be the best component to start with, followed by component 8, because you see a lot of fat clusters are very well spaced out in component 8. Who knows, you would just drop component 8, you'll just go ahead with the component 3, but that secures the most information that you are looking for. This is another idea that we, very simple, no rocket science, but these observations actually coming together, they help you making a very fantastic and accurate and fast algorithm. All right, so with the help of this locality sensitive hashing that essentially finds nearest neighbors, I didn't get into the intricacies of the algorithms, but what it does is it gives us clusters very, very rapidly at that time. Nowadays, people are using this method, but we are the first one to show that you can actually use the hash families to encode single cell data and get much better clusters much faster. We were the ones to do that. And with that, you see at that time, Seurat and Ranger were the methods, go-to methods. Of course, Ranger is much simpler. It just uses a k-means, and k-means is faster, but it gives you stupid clusters, right? This is the default thing that the 10x came up with at the time they released their 10x platform. And this is drop class. We call it drop class because this comes from droplet-based sequencing data that we are trying to cluster, so we call it drop cluster, right? And it scales very, very rapidly. It's in a log scale also, you see pretty linear, right? We didn't continue much in that direction because it was already too fast. But it can be made even faster with some choices of hash families and hash functions. Well, so we got this kind of clusters, as you can see, with the different colors encoding for different PBMC subtypes, and this was the state of the art at the time when we published it, right? This is Seurat. Today, Seurat has improved a lot. They're doing really great, right? And they are also using similar kind of things. They kept improving their software a lot. So I have great deal of respect for them, right? So Seurat is not this today. It was back in 2018. So now we'll talk a little bit about the batch effects. So I think Hamim has already spoken about the batch effects. So if you take a cell line, you do it on 2 different days, the same guy, same machine, you'll see the cells are getting clustered separately, isn't it? So batch effect is the biggest enemy of our progress of science using single cell data today, and probably everywhere. It's just that you cannot do anything. So what happens is people are trying to come up with methods these days, which are just like Botox. You know what is Botox? Right? It makes your skin glow, but also you lose your expression. Right? So it's like Botox. You are getting very neat clusters mixed up, all that are good, but you are losing the small clusters and everything. You have no expression. You have expressivity, means you don't have any information there. So nowadays, integration algorithms have become like Botox. Anyway, but those are the best that we can do. And none of these integration algorithms actually works, let me tell you. This is a sweeping blanket statement, you can argue. But I have tried on multiple data, no single method stands out. In some cases, something works, but you cannot trust any method giving you the right result. This is the biggest problem of single cell biology, which we could not solve as well, okay? We're just showing you one data because this is one of the data we came across. By the way, I'll also talk a little bit about this cell atlas search. What we did was, because we had a very good way of finding out the nearby cell, the similar looking cells using locality sensitive hashing, we actually created a search engine called Cell Atlas Search. And what it did that time was it hosted about 250,000 single cell data and 50,000 bulk RNA sequencing data, and you can query an expression profile and you can get an immediate result. real time, and there was nothing like this at that time. Well, nowadays people are coming with more and more options which you should look at, but it essentially uses hashing as the method, right? We just assign this short bit vectors, and this was implemented on GPU. So this was GPU web server essentially, all right? And the good thing with this was we could very quickly look at how your single cells, let's say you have done a single cell study of simple PBMC. Now you want to compare your single cells with all the single cell data that are out there in the market, right? And you want to measure the cosine distances with them. And you want to use those values as surrogate for clustering and all. Actually, it helps. Projection always helps. So you, instead of clustering the single cell based on their own gene expressions, you can actually use other data as references. You can project on them, and you can get, by that way, you can tease out a lot of small variabilities which are unwanted. It's very hard to explain it here like that, but there are papers talking about this theme. So you know what is batch effect, the same cells getting split, and once you apply batch effect, it gets mixed up, this common story. And we could see something similar happening where when we use cell atlas search to project our single cells, these are the two different cell types, different colors are different cell types, different shapes are different batches. So this brown in two batches, they're clustered separately. So this batch differences are somehow fixed when we are projecting them on using the cell atlas search. By looking at 250,000 profiles, when we clustered them based on how proximal they are with all these 500,000 profiles, in terms of that, when I clustered them, this is much better than clustering them on their face values. Finally, I'll come to rare cell discovery. This is one of the holy grails of single cell biology, and this is one of the reasons single cell biology is done in the first place. You want to find rare cells, otherwise what's the point? You don't want to discover the T cell again, because that's already discovered. People have got many Nobel Prizes on that. Now, how to discover rare cells? These are a couple of examples of rare cells. This is taken from some other study. Pulmonary ionocytes in orange extend through neighboring epithelial cells in mouse airway. And similarly, circulating tumor cells floating around in the blood, they are very, very rare actually, but it's super important to catch them. So that is one of the major themes of research in our lab, which we will not talk about today, by the way, because today we are talking about big data and how big data algorithms can help. So here's an example of what would a single CTC look like in the blood. This, out of 500,000 blood cells, you have only one circulating tumor cell, that rare. No technology can actually recognize them well enough. And just imagine how much of money you will spend to catch just 5 circulating tumor cells, right? If you want to do single cell capture, followed by single cell sequencing, followed by phenotype characterization, it's a huge amount of money you will spend, and still you will get nothing because it's so rare, right? There are, so I don't want to get into CTC biology. It will take me another 15 minutes. So this is one example of rare cells. So there is another comparable method. I talked about locality sensitive hashing, which are basically raising random bars here, random hyperplanes, and putting you in the cubicle and giving you a zip code for your cubicle. That is hashing, right? Locality sensitive hashing. There is another variant of that, and look, this one preserves L1. 5 minutes? Wow. So what locality sensitive hashing does is, what sketching does is it actually draws simple lines along every dimension, and it puts a threshold independently along every dimension, and thereby it generates a hash code. So with that, what we did was by the way, the hashing is always prone to misapproximation, because you can raise multiple random walls, that's okay, but by chance I can be separated from my peer or my friend, right? If you do it just once. So what we did was we did it multiple times. It's kind of a bootstrapped estimator you can think of. So what we did was, these are hash buckets, right? These are single zip code, right, assigned to too many cells which are very, very similar. Like this is a major fat cell type, this is a small cell type, quite rare. So if you do hash them, then for example, if you look at this black cell, they are very few in number, having that hash code, right? So the probability of that hash code is very small. So we can combine these probabilities using Fisher score, and that becomes our statistic. Although it is empirical, it should follow chi-square distribution, by the way. But we don't need that. We can basically rank the cells coming from each sample based on this hash code based approximation of their crowd. So we are basically measuring that after creating all these cubicles, we are just measuring that what is the crowd volume in the room, basically, right? If I draw a cubicle, then how many people are there in the cubicle divided by total how many people were there earlier, and that's the crowding number for that particular cubicle, right? With this principle, we could actually determine rare cells. We could demonstrate for known rare cells, like dendritic cells are also rare. We could show that if you just tune this score, it's just a score in the continuous spectrum, right? It's a real number. So you can put a threshold and you can decide how rare is rare. That's up to you. Like when you increase the number of cells, then you get to see the monocytes. Here you just see the megakaryocyte, which is the rarest of the cells that was there in the data. And with that, we could determine new cell type. I don't want to get into this. This is a published data. We could spot existence of a cell type in the mouse brain, which is a pars tuber cell type. It's very, very rare. It's a subtype of the pars tuber cells, and we could spot it back in the Allen Brain Atlas. Allen Brain Atlas is a Microsoft Foundation project where they have a lot of brain slides and their images, in situ images. Well, so I would stop there. I had just one last story, but I don't think I'll have enough time for that. But the speed up, if you see with this rare cell discovery algorithm, was massive, right? I mean, even today, I don't think too many methods can beat this in terms of accuracy and speed. And this is thanks to the sketching algorithm and the downstream statistical measurement that we created. So this is the real power of the big data algorithms. I did not learn anything, but I learned everything. That is the message. So this is why you guys should take big data seriously, right? So differential expression analysis part is little more statistical. If I have 2 minutes, I can go through this. I can finish. 2 minutes. 2 minutes, yeah. So that was great. Okay, so you know what is differential expression analysis? Between two groups of cells, if you find the genes which are up or down, is differential expression analysis. And we actually made a very interesting, we came across this paper in PLOS One, which, just like normal distribution is universal. It applies on everything. They came up with a discrete beta distribution, which is like this. If you just look at all these boxes, this comes from an artistic picture. So if you take these boxes, you put their rectangle rank, if you rank them by their size, and you put the size on the y-axis, area on the y-axis, and their rank in the x-axis, then they follow this beautiful pattern. And that is always universally true, just like normal distribution. So this can be approximated using discrete generalized beta distribution, which has two parameters, two shape parameters, basically. We could fit this on ranks. You always have heard that ranks are robust, but the statistical power is less, right? So you don't want to use ranks. You want to always use some good estimate. So what we did was we came up with a midway. We said that here is something based on rank. Here's a distribution, discrete distribution that fits on rank, but it is parametric at the same time. So you can find alpha and beta. And you can compare alpha beta using a Wald-like statistic by solving it for using a log likelihood, using a log likelihood method, maximum log likelihood estimator. And you can compare them as part of a Wald test, which includes computation of the Fisher's information matrix and all that. It's not interesting, but it is very, very fast, okay? It's really fast. It is very accurate when we compare with the gold standard data, and it is very fast as well. So here is an example of how fast it is, right? You see, most of the methods, these are like best practice methods even today. MAST is the one that really scales very well. It is published in Genome Biology. And SCD is the first method that came up in Nature Methods back in those days, 2014. And our method grows much slower as compared to any other method that you can think of. And it scales really, really fast. Finally, high throughput omics data are noisy. Hash families are great friends if you can use them, if you understand them. They're simple, like plus, minus, multiplication, and division. Very simple. You will all understand if you are applying them. If you get into the convergence proofs and the guarantees and the bounds, they will become a bit difficult. Not that difficult too. With class 12 mathematics, you'll understand this. One should ignore strong parametric assumptions for dealing with noisy data, just like single cell data. And ranks are more robust. We have shown that in the last description of the differential expressions. And I really promote, I'm requesting all of you who are students here to look beyond your area of specialization, right? So the days of specialized being, so we used to take a lot of pride calling that we are specialized, but that's not going to stay for long. You'll have to be a little bit more generalist to be more useful. The time has come with ChatGPT and all of that. The generalists will be appreciated more because they can see the end-to-end. So you'll have to look at least 2-3 other fields if you want to do transformative science. Otherwise, you'll be just circling within a box. So these are the collaborators, students, and funding agencies which I'm grateful to for this works to take them to conclusion. And this is IIITD, where I belong, and you can follow my lab on Twitter. I can take a couple of questions.
So I have a question here. So any hash-based method is sensitive to the dimension of the hash, right?
Yes.
So how do you decide which one would you choose?
We try out a very good question. Very good
shot
question. That's empirically decided, basically. Because we try out a few things, but we have typically seen this between 10 to 20 hyperplanes. You can approximate your data fairly well.
Okay.
But that you can always try because it doesn't take any time.
Yeah, I mean, in fact, it can be done parallelly also. Because
it can be done parallelly on the GPU also.
Massively parallel you can do.
Yes.
That's why it's so fast.
Yes. These are matrix multiplications in the end of the day.
But the size is something that...
Size is I would say yes, you are absolutely right, but I think it's between 20 to 30, 40, it gives it's decided empirically, frankly speaking. We had to tune it.
But still it is better than the others.
Still it is much faster and accurate also.
I see. Okay. Thank you.
And no estimation going on. Yes.
As a follow-up question to Professor Debnath, along with dimensionality, I think LSH works best with uniform data.
Yeah.
So even with
LSH
bootstrapping also works best with uniform data.
Okay.
So even with bootstrapping, I think there will still be a bias seen. Has such a bias been seen in the experiments?
So you are saying LSH works on a particular kind of a data, uniform data?
I think it works best when the data
it works actually, frankly speaking. It will only work when the data is not uniform. If there is a structure in the data, then only you will expect cubicles to be meaningful, right? Like here, there is a structure in the data. You guys are sitting here, these guys are sitting here. That's why there are chances that if I draw a line like this by chance, then you guys will be properly preserving your structures. So it works only when there are, according to my simple understanding, it works when there is a lot of structures in the data, a lot of clusters in the data. But I think there is a different thought process behind this question, so which we can discuss later.
Thank Debarka for very energetic talk.
Thank you.